Assalamu alaikum, my dear viewer. I'm your host Fahad and you are watching my YouTube channel Lecture with Fahad. Today we will discuss the techniques used in cell biology. What is cell biology? Cell biology is the study of cell. It's called cell biology. It is also called the cytology. Here we will study different techniques used in cytology. We will discuss three main techniques. The one is staining. The second one is staining fractionation. Number third one is tissue culture. We will discuss these three main techniques used in cytology. The first one is the staining. As indicated from the name, staining. Staining is taken from the word stain, which means color. So it is the process of coloring. So when different organelles of the cell are colored, it is called staining. What is its purpose? Because many of the organelles are transparent, we need to give some stain to it for better study. Staining help in increasing resolution power. There are many types of stain. The one is the vital stain. Vital stain. It is that stain which doesn't harm living cell. This is called vital stain. There are many other bases on which we can divide stains. For example, primary stain. The stain given first is called primary stain. Number second is the counter stain. The counter stain is that stain which is given after the primary stain is called counter stain. There is other two types of stain on the basis of durability. One is the permanent stain. This is that stain which cannot be removed after adding second stain or counter stain. It is permanent. That's why it is called permanent stain. Number second, temporary stain. Temporary stain is that stain which can be removed after adding second counter stain. Fractionation. Fractionation is the second technique used in cytology. Fractionation is taken from the word fraction. Fraction means parts, or dividing something in part is called fractionation. But in biology, when cells are divided into different parts, this process is called fractionation. And when this process is done with the help of centrifuge machine, when this fractionation is done through centrifuge machine, then this process is called centrifugation. There are two main points in centrifuge machine, that it has spanning ability. Second, it can be set at either speed, low speed, medium speed and high speed. Procedure of centrifugation. First number step is take the tissue which is to be studied. Second number, grind the tissue in the incubator machine. After grinding the tissue, we will get homogenate. Homogenate is that substance which is made from the grinding of tissue having uniform composition throughout. After grinding, this homogenate is poured into centrifuge machine. So set the centrifuge machine at low speed at first, so different part will be start to separate. After slowly and gradually we will increase the speed of the centrifuge. So first cell will start to separate, then nuclei will separate, and then organelle will start to separate as we increase the speed, and finally ribosome will separate. Then we will easily study every organelle very easily. Third number technique is tissue culture. It is that procedure in which a part from an organism is removed and thrown into such like plant or any organism, especially in plant, in a medium called agar, is called tissue culture. It is a type of cloning, special type of cloning, and it gives 100% identical result, except mutation which may occur during the process. Now I am going to tell you about the procedure of tissue culture. Its procedure is that select healthy part of the plant, especially apical tip, apical bud. Why we select apical bud for this purpose? Because apical bud consists of a primary meristem in which the growth rate is so increased. That's why we take a healthy part of the plant, then cut the second number step is cut the healthy part of the plants. Third, pour that desired healthy part and the medium. In the medium, this is such medium in which optimum temperature is there, optimum pH, all the condition is optimum. So it will be cell division will be start. After it change into callus, then we will apply and edit hormones necessary for the growth of plants. Such is cytokinin. Cytokinin is that hormone which facilitate root growth. Cytokinin help in the stimulation of root formation. We also increase the growth of shoot through auxin, which is another hormone which help in growth of shoot auxin.